ARID1A and ARID1B, along with ARID2, are members of what I will call in this talk the BAF complex for BRG1 BRM-associated factors. It's also a mammalian switch-SNF chromatin remodeling complex. We found that about 11% of the neuroblastomas in our initial study had mutations in ARID1A or ARID1B, and these were associated with early treatment failure. Death from time of diagnosis at about 1 year, in contrast to McGann-amplified tumors, which was closer to 2 years. You can see that the complex has many member names, BAF and their molecular weight, but that was too simple, so they've all been changed to use the new terminology of SMARCs for switch-SNF matrix-associated actin-dependent regulators of chromatin. So that's the terminology I'll use going forward, but if you have literature confusion, that's because of that. There's for instance, SMARCB1 is known in ATRT as INI1 and SNF5 and BAF47, so the nomenclature gets unwieldy. The complex itself is a 2 megadalton complex. It has about 15 members, core members. It's ribosome sized. It's very large. It's about 12 times larger than the nucleosomes it repositions. The interesting factors about core membership is that they are entirely dedicated to the complex. They don't really have a monomer life. And these complex members are also very tightly associated and resist denaturation up to over 2 molar urea. So there's 29 BAF complex genes, and that's because there are 9 positions which can be occupied by one of a couple of mutually exclusive paralogs, and that provides, theoretically, for over 1,000 possible complexes by combinatorial inclusion. And this complexity allows biological processes to be driven specifically, and if we look evolutionarily, the increasing complexity of BAF complexes mirrors neurodevelopmental complexity. I point out here the 9 spots with the changes of the different paralogs, and we'll come back to that because it has a role to play in neural differentiation. So the BAF complex became much more interesting to cancer biologists when the frequent sequencing studies of different diverse cancers were being published, because about 20% of all human cancers have been identified to have mutations that are either activating or inactivating in members of this complex. I'm highlighting some of them here. The bold ones relate more to pediatric cancers, obviously the teratoid rhabdoid tumors with the SMARCB1 loss of function, and synovial sarcomas with the translocation of the SS18 component, and neuroblastoma highlighted in red with the ARID1A and ARID1B we identified, but I've also starred some of the other members of the BAF complex that were found mutant in some of the other neuroblastoma sequencing studies. So this is a summary of our recent extensions into 72 neuroblastoma cell lines, 69 unique cell lines. You can see that we've used a variety of techniques from whole genome sequencing to cancer panel. I'll point out that the Foundation Medicine Cancer Panel we use does not have ARID1B on it. We've also used a targeted capture for ARID1A and ARID1B, and some data from the Broad cell line, cancer cell line encyclopedia. And below I show the mutation spectrum for the ARID1A, B, 2, and SMARCA4 or BRG1 components. And what was interesting to us is that 9% of the tumor cell lines had a biallelic ARID1A inactivation. ARID1A maps to the 1p35 region of common loss in neuroblastoma. Because it maps there, we know a large proportion of these cell lines also harbor a monoallelic deletion. It's deleted in about 80% of tumors with deletion, and at this point we don't know if that haploinsufficiency has any functional relevance. ARID1B mutations were seen in about 18% of tumors. We never saw a biallelic inactivating mutation, but the mutations we do see for the most part are clearly inactivating, and often accompanied by what I would call compound hemizygous mutations, again the significance of which is unknown. So overall, with some incomplete data that we're continuing to mine and extend, for this panel of cell lines, 26% of the neuroblastomas had mutations in one of these 4 genes. and there was some primary tumor data at a morning plenary session from Gudrun Schliermacher's lab looking at primary tumors, and we've done some primary tumors as well and find a similar frequency. We had some initial work done as part of our principal published sequencing effort, and we collaborated with Mike Dyer and Sarah Federico to look at primary tumors in an extended set, looking at ATRX, ARID1A, and ARID1B. And the take home is that we saw, again, with the inability to call yet deletion events only, intragenic deletions we could call, but not whole gene deletions, about 3% of high-risk tumors had deletions or alterations in these genes. So what we're doing in the lab is trying to understand what's downstream of these mutations. So this is a large complex chromatin remodeling complex that positions nucleosomes, alters genome accessibility, and changes the output of a cell. I would say it also changes the cell state. It affects differentiation, potency, stemness, identity, whatever term you like. So it's a very important master regulator of cell state. I'll show you some data on the BAF complexes in mutant and wild-type neuroblastomas. The work that I'd like to be showing you in the future would have to do with some of the other programs, which I'm not ready to show, which have to do with binding sites of these BAF complexes in the genome and whether mutation affects them, whether the transcriptional output is altered in any predictable ways, and in particular the dependencies of mutations in this complex using a CRISPR-based screening approach. So just nuclear extract input can simply show us that for ARID1A, if you are biallelically inactivated, you have no ARID1A. The ARID1B hemizygous tumors have what appears to be a deficit in ARID1B, and the only other finding in terms of generic nuclear extract input would be that the BRM or SMARCA2 gene seems to be underexpressed in mostly ARID1A and ARID1B mutant tumor cell lines. But we've yet to investigate that further. Looking at stability on a glycerol gradient, this is a wild-type cell showing that about the 2 milladalton, megadalton, complex in fractions 13 and 14 on the glycerol gradient contain most of the core members. When you're hemizygously deleted for 1A, you basically form a pretty normal wild-type complex, surprisingly to me. When you're 1B deleted, you do also, although the stoichiometry is off, and we think you have fewer of them because of the lesser 1B quantity. If you are ARID1A homozygous inactivated, I'm showing two examples here, you again surprisingly create most of what looks like a normal complex. You get some shifting to lower molecular weights because you're missing a 250 kilodalton member, but except for the absence of ARID1A, you can continue to put the core complex together. And basically we showed by immunoprecipitation and silver staining with antibodies against ARID1A and ARID1B, what was known previously is that these complexes have mutual exclusion of those two family members, but that the complexes otherwise, with the exception of the changes in the abundance of 1A or 1B itself, look fairly wild type, which tells us that maybe these are late members nucleating some activity, but not necessarily required for complex formation. So the other thing I wanted to talk about is the role of the BAF complex in mediating neural differentiation, because we talked earlier about the combinatorial switches possible. And in this pathway, there has been a nicely worked out role for a switch of these three members, PHF10, SS18, and ACTL6A, to their paralogs. And that is coincident with change from what we call a BAF complex that exists in a neural progenitor, or an NP-BAF, to a neuron complex, which drives neuritogenesis genes. And I'm showing to the right a panel of the switch from SS18 BAF53A, which is the neural progenitor BAF, towards a later point in mouse cortical development where you've switched membership to the SS18L1 ACTL6B or neuron BAF. And we see evidence of this in the neuroblastoma cell lines, that wherever there's those sort of mutually exclusive members, it looks like the expression of the NP member exceeds that of the neuronal member. Looking at the data sets available through R2, the COSAC 649 neuroblastoma data set, we asked whether it looked like there was co-regulation of these three members in the neural progenitor BAF complex, and it does clearly show evidence that these are co-regulated, directly correlated with each other. Similarly, within the NBAF complex, the three components that have been switched out are now co-regulated as well in that large data set. And conversely, when we compare the NP-BAF members exclusively against the NBAF members, you get an inverse correlation by and large for those members we had data for, suggesting that you tend to exist in one state or the other. Looking at the downstream neuritogenesis genes, we see the same thing. The NP-BAF complex is indirectly related, while the NBAF complex is directly related. And we also see, using this available outcome data sets, the Versteeg 88 and the Seeger 102 that many of you may know, that the changeovers in these genes correlates with outcome. I'll just quickly say we're looking for biomarkers of activity that would give us a better handle on what's oncogenic when you have mutations here. The only thing we've looked at so far is this anaphase bridging assay, which tells us that ARID1B haploinsufficient tumors have much more anaphase bridging because of the effects of the complex on TOP2-mediated DNA relaxation. Surprisingly, ARID1A null cells seem to be wild type, at least this aspect of BAF biology. So the summary, I will just hit the high points. We think mutations in the BAF complex are common. We believe that biallelic inactivation of ARID1A strongly supports it as a 1P tumor suppressor gene, a classic Knudsen biallelic gene. The prevalence of these mutations is higher in cell lines, but significant in primary tumors as well. There seems to be co-regulation of this complex with differentiation held up seen in neuroblastomas with more aggressive behavior and poor outcome, and we have a lot of work to do to better understand